Hello everyone, I'm Yixuan, and I'm excited to share our project, Multi-Omics Integrated Cancer Analysis. Our goal is to build a scalable cloud AI system that integrates multi-omics data to predict cancer types, identify biomarkers, and perform survival analysis. Our key innovation lies in fusing RNA, DNA mutation, and clinical data with advanced AI techniques. The core challenge was managing this massive scale while maintaining biological signal. Then for data, we use the TCGA Pan-Cancer Atlas on GCP BigQuery, covering over 11,000 samples across 33 cancer types. We processed a massive volume of over 50 terabytes of raw data and finally distilled it into 500 MB of clean data. The multi-omics data itself shows variety, well-distributed technologies like BigQuery and Dataproc ensure efficient processing. Then moving on to our data engineering pipeline, we started by aggregating heterogeneous data from BigQuery, covering clinical, RNA, and mutation domains. Then we process this through a three-step pipeline. First, we select our cohort, filtering for the top 7 cancer types. Second, we handled feature engineering, which involved pivoting RNA data and standardizing mutation IDs. Finally, we integrated these data sets and applied strict quality control to remove about 1,100 empty samples. The final output is our master matrix. It contains over 2,700 high-quality samples with nearly 40,000 features, combining both RNA and mutation data. And you can see the quick review of our master matrix. Then to handle the high dimensionality, we used an autoencoder compressing input features by a roughly 40:1 ratio into 100 latent features. Using t-SNE visualization, our data separated into 6 distinct clusters. You may notice an overlap at the top here. This represents 2 different types of colorectal cancer. Given that they are high biological similarity, this clustering validates that our AI captured the correct underlying biology. Moving to our tasks, we first develop a classification system using XGBoost on these 100 latent features. For results, we achieved nearly 90% accuracy. The confusion matrix heat map shows strong diagonal patterns, proving that these 100 dimensions contain sufficient information to distinguish cancer types with minimal error. Further, we identified the 20 top driving genes. By filtering the 2,000 most variable genes, we extracted feature importance scores to provide interpretable biomarkers for further validation. Next, we conduct survival analysis and gene decoding. First, we plot KM curves to check data quality, and it shows that our dimensionality reduction perfectly captured the clinical characteristics. On the left plot, you can see this purple line. Purple line represents the breast cancer. It has the flattest slope, indicating the highest long-term survival. On the right, the gap between the blue OS line and orange PFS line represents survival post-progression. For breast cancer again, you can see here this wide gap confirms that even after relapse, patients can survive for a long time with effective treatment. Next, for modeling, we treat 2 Cox proportional hazards models, achieving a stable C-index of both roughly 0.66, indicating robust prediction without overfitting. Then to decode the biology, we used a tornado plot to identify dual threats. features that both driving dice and progression. By correlating these features back to the raw genes, we found that AI automatically revealed some underlying mechanisms of cancer biology. For example, our strongest protective factor, latent 85, correlates highly with pol mutations. In biology, these mutations trigger a strong immune response, leading to a better prognosis, exactly as our model predict. Finally, we assign the risk score to each patient. In the survival plot, the high-risk and low-risk groups separate clearly, showing that our model can support patient risk stratification and potentially inform follow-up intensity and treatment planning. We also validate the robustness of our classification biomarkers. After controlling for cancer types, POU3F3 and IGSF11 remain significant prognostic factors across cancers, suggesting they are not just subtype markers but have pan-cancer prognostic relevance. Here's a quick review of our system. The entire process is fully integrated with GCP, forming a complete prediction to interpretation to validation loop. This highly reusable framework offers substantial value for downstream biological discovery and real-world clinical decision-making. Finally, in conclusion, first, for technical performance, our unsupervised representations provide strong and stable prediction for both classification and survival modeling. Then, for biological discovery, via gene decoding, we connect predictive signals back to interpretable biomarkers and plausible biological mechanisms. Finally, for clinical utility, together our project enables an end-to-end multi-omics framework for outcome prediction, risk stratification, and biomarker discovery, bridging model performance with actionable and explainable clinical insights. That's all. Thank you.